Hi everyone, welcome to BDBIO. My name is Sanchit, I'm from Intel Labs. We are going to start the first session, and I think we have a very exciting couple of talks in this session. The first talk is going to be on reimagining drug design in the era of artificial intelligence. So let's welcome Professor Arijit Roy, Dr. Arijit Roy from TCS for the talk.
Yeah, good morning everyone, and first of all thank you to all the organizers for inviting me here. So as you can see from this title of this talk, that I'm going to discuss about the drug discovery and drug discovery in the era of generative AI. So I will try to share with you our learning. I think we are working in this very particular field for the last 4 years or 5 years. So I will share with you what we have learned, how we can accelerate the drug design. So it's a big process, drug discovery. So today what we are going to what I'm going to discuss is more about the early stage. So that's the design part. So let us see. So before going into the details, let us first try to understand why we need newer methods for drug discovery. As you can see from the left part of this slide, you can see there is a huge R&D cost and time in order to bring a new drug to the market. So of course the pharma want to reduce the cost and time. One major problem is the late stage attrition. You can hear me clearly, right? Okay. They also want selective and non-toxic drugs. And due to the resistance issue, they also want drugs with new mode of action. And of course, it is also important for patient, the affordable drug, quick access the drug quickly. None of this possible with this huge time and cost. So our interest is on the early stage drug discovery that I mentioned, from identification of hit to optimization of the lead molecule. And it roughly takes 5 years and 500 million USD. So we want to aim to improve the success rate with AI-based drug design, which can bring benefit to both pharma and society. So now let us see beyond time and cost. So what are the pain areas in the overall drug discovery? As you can see that one of the major problem is that so they are always limited to the number of molecules that exist in that library. So what I will try to show you that how with the help of generative AI, we can explore a very, very large chemical space which no one has explored ever. So here the AI, the generative AI gives us opportunity about how we can explore this big chemical space. The other problem is that if you think that once a hit molecule is identified from a library of molecule, in the next few steps, people try to see that how they can optimize various properties, drug-like properties, so that they can get a good molecule, good hit molecule, or rather some lead molecule. So during this process, various properties needs to be optimized, like toxicity, solubility, or various admit properties. Now since this is a sequential process, so maybe after 2 years, 3 years, 5 years, people find that one of the property is not working. And that's the reason for the late stage attrition. So in that case, people need to go back and start it again. So what I will try to show you that we can also predict many of these properties through various predictive AI. Now, instead of screening approach, what we can do, we can, with the help of generative and in combination of generative and predictive AI, we can actually sample from this chemical space, that the huge chemical space that is available through a automated process, where many of these properties can be optimized on the fly. So there is less chance of late stage attrition, and that's what I'm going to show you. So this is also the way I have organized my talk. So first I will try to tell you various generative AI model that we have developed and how it can help us to explore the big chemical space. The next part I will talk about how we have developed various predictive AI methods and how in combination of generative AI, it can reduce the chances of late stage attrition. And I will also not talk much about the lead optimization today, but I will show you that how we can get the explainability out of this AI model, which can help much to the medicinal chemists. They can understand what is the importance of various fragments so that they can change those fragments during their lead optimization to get a better molecule. And since many of these molecules are new, so their synthesis can be challenged. Synthesis is anyway challenged. So I will also try to show you how we can overcome with the help of AI. This is largely what I will talk about. So this is largely, these are some of the methods that we have developed, and I will go about that. Some of this predictive, this generative AI method, we have number of generative AI method. I'll go into details about one of this method, possibly this one, and I will try to show you that we can, there are various different conditions one encounter during the drug discovery. So there can be various inputs. So some of these various scenario we tried to think, and then we have also number of predictive AI model, and these are actually combined with generative AI model to sample from this large chemical space. This can improve number of limitations of the current method mainly through multi-property optimization. And I will also talk about the explainability. So what is shown in this figure at the left hand side is that there is a huge chemical space available, which is 10 to the power 60 or more. People have shown that through combination of various fragments. Now we have never explored this chemical space. So when pharma companies work with this library, so it might be few million, now people are talking about ultra large library, but that also limited to billions. But this chemical space is huge. So we want to explore this chemical space. We want to sample from this chemical space. So then we have opportunity to get much bigger chemical space, which may help us. So how machine can understand, how generative AI can understand. So a molecule can be represented in various ways. So 3D, XYZ coordinate, and it can be also represented through 1D. So that representation known as the smiles. So one can represent this molecule through these smiles. So it has a well-defined grammar. You can also represent the complete molecule, and it has a unique grammar, and each molecule has a unique grammar like smiles. So when you put it in this textual format, the existing NLP algorithm can learn this grammar. And when it learns this grammar, it learns from various molecules, all the molecules that is available, let's say in a chemical library like CHEM or zinc, then it has opportunity to learn that grammar, and then it has ability to create new molecule and sample possibly from this chemical space. At the same time, we need predictive AI model. So this predictive AI model, like the bioactivity, that is the most important part when people do, experimentally virtual screen, they try to see that how well it can bind with the active site. So we can also build those models, and we can also build models like solubility, various ADMET properties. And when we combine both of them, we can sample in a focused chemical space, which is very specific to the target molecule. And that's what I will show in the next step through the method which is known as reinforcement learning, which is used at many places. For example, we have a generative model. So these were very initial models that we used. We used a recurrent neural network. So right now we use everything like transformer to other models as well. So it learned the grammar of the small molecule from a library like zinc or CHEML, where people deposit these molecules, and it in the smiles format, and it tried to generate new smiles. It has now, since it has learned the grammar, it can now generate many new smiles, and that is passed to the predictive model. This predictive model can be bioactivity, or it can be one, it can be multiple. So then it tries to predict whether it is specific to that target. So if it is passed that, if it gives a positive reward, the whole model is rewarded, otherwise it's penalized. So what's happening in that way, there is a on-the-fly optimization. The model is automatically learning that I need to produce some molecules which is very much target specific, or I need to focus at the chemical space. Example, if it is predictive model is bioactivity plus solubility, I need to focus at that chemical space which is target specific, plus I also need to support the solubility, otherwise I will be penalized. So it's gradually, there is a on the fly optimization so that it's moving towards that chemical space. So that's what I explained here. So we started with a chemical space. So actually this is much smaller. The actual chemical space is very huge. So my initial aim was to explore that big chemical space, learning the grammar of the small molecules so we can generate many novel molecules. But what happens that sometime medicinal chemist has some idea, I mean they have tested some fragment or molecules like this. So we already have some information, or we already know some molecules which are active against the target. It's not perfect. The IC50 is not great, but we know that those are working, or it might be a small fragment. So what we can do, so we have learned to explore this chemical space, but I don't need to explore the big chemical space. I only need to focus at a particular chemical space, and that these molecules are helping. So there is a method which is also called transfer learning, which is actually helping me when I learn these molecules, when we show the model that, okay, I need to focus at that particular chemical space, then it will focus only at that particular chemical space. So, and then through reinforcement learning that I showed in the previous slide, it will try to optimize various properties and go to that particular chemical space and produce much more protein molecules. So these were very initial works. I believe that we don't had much contribution here. So this is exactly what we started. So we had a large library CHML, like 1.6 million molecules. We had a pre-trained generative model, and then we fed the model with a target specific data set. And then we have that reinforcement learning framework, and we tried to generate property optimized molecules, and those properties were not considered. We use additional filters, other filters to get the final molecules. So I believe our only contribution in this particular works, this ligand based method, was that we showed that how we can get a target specific data set. But there are other works later we did where we feel that we have a much contribution to that field. I will come into that details, but this is the only work I want to go into little bit details. So we have a pre-trained generative model. So it initially tried to learn the grammar of the small molecule from library like zinc or CHML. So we need to see that whatever it is producing, whether it is producing good molecules. So we try to see log P, molecular weight. So actually it's within the range. So whatever it has learned, it also tried to produce. At the same time, we looked at the guacamole data set, and we saw that the molecules were 96.6% chemically valid. And also it has a high internal diversity. It's not just producing the same thing that it has learned, also it satisfy the property. But right now I think with the latest model it's almost 100%, we can produce 100% model. So here what I'm trying to show that the predictive model has produced good molecules. And then we also generated so we have, for this particular work, we have done an in silico validation. That means that JAK2 is a very popular target. We took the existing inhibitor of JAK2. So this is Janus kinase 2. We took the existing inhibitor of JAK2, and but we kept it separate. We never showed those things to our model, and our aim is to see that whether we can reproduce some of those molecules. And but we created a training data set from the homologous proteins of JAK2, and we showed that we can also do it from other source by just docking the molecules against the JAK2 inhibitor, and created such model for the transfer learning, and we generated some molecules. And we show that there are, we generated at least 310 molecules which had a high similarity, and this similarity was captured in terms of Tanimoto coefficient. I'm not going into details, which showed that the molecules are quite significant. And what it showed that here we used as a bioactivity docking score, but later on also we have a model where we can calculate the IC50. And from the training data set, the initial data that we collected during the reinforcement learning, what we show that there is a significant shift in their predicted docking score. And So that's where we stopped and tried to sample from that particular epoch and try to see that what it is producing. And what I showed here that it could produce molecules in good number. And we also could look at various fragments that were the most frequent fragments. And from the training data set to the validation data set which was separated, we tried to see that how those changes. And I mean, the only important, don't look at these numbers, but only important thing here is that what we see that during reinforcement learning, there is a significant change. So actually, we just wanted to show that during reinforcement learning, model has learned based on this bioactivity model and other things, and actually it is producing fragments which is more target specific. So that so during reinforcement learning and transfer learning, actually those things are happening. So reviewer also asked the question that how you are sure that you are producing molecules which are very specific to JAK2. So we went back to the patent because JAK2 is a well studied protein, and we tried to look at various fragments. So these are the most popular fragments from those patent literature we collected, and we tried to show, I mean, that whatever the molecules that we have generated also contain many of these fragments. So in that way, we tried to convince that because also the selectivity is a question, because JAK2 is a part of a homologous family which also contains JAK1, JAK3 and TYK2 other proteins. So how it is selective? So that question we also answered, and this is one of the most a very selective JAK2 inhibitor. What we found that some of our molecules are very similar to them. I mean, only difference is possibly at this position and at this position. But we also got those type of molecules, and we never showed the model to the any of these molecules to the training model. So other thing I was saying you at the very beginning is that late stage attrition is a very big issue, because people try to, once they find some hit molecules, they try to optimize various properties in a sequential way. So we try to answer that from a protein of a central nervous system, how we can overcome those, reduce those chances of late stage attrition. So we have taken a target 5-HT1C from central nervous system. So usually for central nervous, any target of central, if you are designing molecule against a central nervous system target, so usually people try to see at the molecular weight is usually small compared to the other therapeutic area. So blood-brain barrier permeability is one important part what people need to consider, because that's one of the challenge. And also the solubility, the log P is somehow control this blood-brain barrier permeability. And I mean, what we tried to do here is that we include all this property so that the generative AI model can focus at a particular chemical space where all these properties can be simultaneously we can optimize. How much time we have? Okay. So we are also interested about the explainability of the model, and our contribution is that we could do it at the fragment level. So most of the methods were at the atom level. So what you were saying, I just want to discuss one particular example. So these two molecules, all these pairs of molecules are known as the matched molecular pair. That means that they are identical, but they have only difference at one position, that is shown at yellow, with yellow. And due to this property, there is a complete change in their function. So for example, this molecule is blood-brain barrier permeable, but this molecule is not. So we could successfully say here that, okay, this molecule is blood-brain barrier, and we do it at the fragment level. We can show that from this explainability model can tell that why certain fragment is good in for example, crossing the blood-brain barrier or not. But we also had some unsuccessful case, for example, these cases. So we try to look at it very carefully. So usually people say that if there is a hydrophobic group, then there is a higher chance of blood-brain barrier permeability. But if this molecule is blood-brain barrier permeable, but experimentally it showed that this particular molecule is not blood-brain barrier permeable. So we were I mean, we just asked question in that particular paper that there might be something wrong in those data because there is the addition of hydrophobic group here. So if this molecule is blood-brain barrier permeable, most likely this molecule also should be a blood-brain barrier permeable. And similarly, so these are some of the ligand-based method, but then we are also wondering that how we can overcome the challenge of the low data scenario, because although we are talking about the big data, but ultimately the challenge is the low data. I mean, in most often we encounter scenario where we need to work with a very low data scenario. So most often, most of the generative AI methods are ligand based. So that means that you have a small amount of data, it can be very small, and people try to optimize that further to get a potent molecule. But there are also scenario you are starting it for the first time. So that's where our structure based method come into picture, and which actually work in a low data scenario. We have trained this model with the existing protein structure and the small molecule. So we separately train them. This is a graph neural network based approach, and the smiles based approach. We concatenate that. We train the model both on the protein and the protein structure and the small molecule. So basically the model try to learn what are the amino acid residue, what type of amino acid residue, what is the volume. And once it learn it, given a new molecule, it try to predict what should be the, based on that condition, what should be my new molecule. Again, we validated it on JAK2 and TYK2. So I'm not going into the details about this, but one important part, I want to show you the explainability part. So here the graph attention network told us which were the 8 binding site residue, and most of them, at least 6 of them, were part of the active site. We wanted to know what are the residue that got more importance during the molecule design. So what we saw that there were some hydrophobic residue and there were also some charge residue. So what's happening is that I mean, it's not true for all the cases, but at least 70% cases we observed when we tried to closely analyze those results. We saw that where there are charge residue, the molecule that is designing, the part of the molecule that lies over there, it somehow get more interactions, hydrogen bond or other interactions over that thing, and the part in the active site where there are more hydrophobic residue, the molecule also has hydrophobic residues. It's not true for every cases, but at least 60-70% cases we have seen this particular trend. So I'm not going into the gene expression based method we got, but there is one method I'm particularly interested because the synthesis can be a challenge. So in this particular method, which we call as a synthesis aware method, we start with a small fragment. So experimentalists often do a fragment based screening. So that can be our starting point. Now we have a USPTO data and around few million data of various type of chemical reactions like Suzuki coupling or, so we try to see that if this is the fragment, what are the part that can be added to one of this template, reaction template. And there is also purchasable fragment library like enamine or so. And we also try to see that how the other part of this template can be added. We already mapped that. Now we have this is a little bit different method compared to the other methods that we developed, because here we are doing a Monte Carlo tree search. We start from this initial fragment and we try to grow many child nodes over there, and we also try to give a reward in terms of a drug target affinity. So that means we try to predict the IC50 instead of the docking score, and gradually whichever child try to get the most reward, we try to select that, and we have an example of protein kinase A on that regard. So we are trying to do a lot of work on the RNA, maybe sometime else I will talk about it. So we have, because small molecule against RNA is really a challenging field, so we started at the very beginning by generating a data set, and druggability is also issue because RNA is a more flexible, and at the moment we are trying to work more on the small molecule drugs, I mean, how we can do it. I mean, it's still an ongoing work, but this will be one kind of a first thing that we can do. The last thing I want to discuss is that we are all talking about big data, but whatever we have seen that data is the issue. And as a community, I think what we need to do, we need to work more on data extraction. So we just last week we have released one data on, for example, the kinetic data, I mean, that is available from Brenda, and we have also generated certain three-dimensional structure. But we have also done a lot of thing with the chemical reaction, chemical reaction data. For example, this is one such data extraction. So what is happening here that we have taken LLM, it can be GPT, it can be Gemini AI or anything we have tested. So we are giving, for example, we have a method which can tell that which are the reaction containing paragraphs. Now we are supplying it to the LLM model and asking it to find what are my reactant, what is the amount used, and what is the product, and what is the yield, and what is the solvent. And we saw that it is very good at it, but there are multiple other steps because stoichiometry can also be issue. How many molecules and we have used. So we have just released, I mean here in this particular work we showed that how LLM can be used, but we are also in a process to extract a bigger data because that I think can be helpful for the community. I think with that I will just end here. Thank you for, and this is the team. Thank you all for your kind attention. Thank you.
It covers so many different
Yeah.
problems in drug discovery. We have about 7-8 minutes for questions.
Yeah.
So stereochemistry is an important issue in any drug discovery, but using smiles often does not take care of those aspects. So
No.
what are you doing on?
In this
regarding
work we have not done, but you can take care about the stereochemistry with smiles. You can.
Okay. So
For example, right now I was talking about this data set. I mean, we are trying to predict the KCAT, KM. So over there we are using, so it's possible. Here, if we do it, the problem will be there will be multiple things will be generated.
okay.
And even from the data where it is learning, like CHEMBL, or the problem is that we don't have that information. So it might be limited for certain cases. It might be there, for certain cases it's not there. So that's why we are not taking care, maybe in the later stage, I mean when we are discussing much, doing some QM, I mean further whatever we have shortlisted. So we are trying to look those aspects at that stage. So the idea is to quickly give idea to a medicinal chemist so they can find the right thing, they can play around with all those things and come with a solution. And then we can check out further from those top 10, top 20, we can do further work on those things. So in this work we have not considered, but it's possible to add stereochemistry in the smiles format as well.
Okay. One more question.
Yeah.
So in the ChemBell database that you typically use to refine your generative AI procedure, we don't find many natural products and their input integrated into these databases when we know that those are kind of an information that would probably be useful to eliminate many of the toxic or other components that typically cloud your drug discovery, right? So what is being done on these aspects? Because as you say, there's dearth of data, but there's a lot of data on natural products. But
Yeah.
I don't see that there is any convergence of these two thoughts.
So indeed there are many data for natural product like coconut. You must be aware about coconut database, which is now one of the. So we tried to look into those databases. The problem is that we have not solved it exactly. I mean, we thought about it. The main reason is that if you look at the Lipinski rule, I mean, which define that what should be the drug-like properties. I mean, there is a range like the molecular weight or other things. That is completely different in case of natural product, which is much bigger, there are many hydrogen bond donor, acceptor. So that is one reason, I mean, we are thinking, I mean, how to deal with that, because I mean, then we need to train the model in a completely different way, because these molecules, if you look at the chemical space, is quite different from the other synthetic drug-like properties. But it's a good question, and maybe at some point one need to look into that, because that's the area. But what I am showing it that we also had done this gene expression based work. I mean, the model is trained on the L1000 data. I think many of you are aware about that. Not a great data, because we spent around 6 months on this data. But what is important is that this data is trained on small molecule. For each small molecule, what should be the gene expression, and that they have done for multiple cell line. So one work we did where we took all the natural product, and for at least 3 cell line, because although there is a lot of data here, we try to predict some of those natural products gene expression data at least for 3 cell line where there were more data in the L1000 data. So that is one work. But I think we need to do more. I do agree with you, and maybe at some point we or someone else will do that. Yeah, go ahead.
So when you talk about RNA as potential drug targets, so I am assuming multiomics data is like involved.
Can you repeat?
Multiomics data is involved when we talk about RNA.
No.
No?
No.
Okay.
No multiomics data is for the RNA things. Because the structural data is also available. Because, and what we have collected from PDB and other sources, and we also generated some data in this particular board. This was done with Professor Gromiha at IIT Madras. So we have also collected these data. These are crystallographic data. And we have also generated some data on the top of it where the kinetic data is there, but it's not there.
Okay, so currently we are looking at the available space of structures, right?
Yeah.
So for potentially new targets, we might need to go for multiomics.
We need to think about that because one question is that RNA is flexible.
Yes.
So what should be your druggable site? Because I mean, what we have seen, if you look at literature like HIV TAR or some of those RNA, I mean, multiple ligand can bind with different conformation. Or people also have shown that in a RNA conformation, a single ligand can have multiple conformations. So that's one of the problem with the RNA due to its flexibility and other things. So that's why this might help. But I think with this low data, we need to do more. I mean, it's not accurate. I cannot claim it's accurate. So
no, because we have the splicing and all these things are also there. So we. I think
but what is important here is that if you read this statement, right now we have only 700 human proteins which can be druggable. So this gives us opportunity to explore a bigger molecules for which or targets which we can consider as our probable drug target. So that's this space is giving.
So that might future studies might also increase the potential of a like say coding region to be a target in different conformations.
It is possible. I mean, it's an aim. I don't know whether it's possible or not.
Okay, sir.
I mean, it's a distant dream, I should say.
Thanks, sir.
I mean, some of these things possible. I mean, don't believe that AI can do anything. That is the first thing I would say. Many people believe that. I mean, only it can do something based on the data.
Sir, yes. Sir, continuing Professor Pal's question, using the natural product database, is there any predictions of the specific property, especially on specific pathology, like for example, using the oncogenes and all, no? I'm talking about especially the malignant conditions. So, is it not possible to actually apply prediction of these molecules using the natural product database?
I don't know. That will be my first question. But I think for any prediction, we require some good data so that the predictions is right. I believe that those type of data is limited for at least for the natural product.
No, with the available limited data itself, because as I am working with the natural products, especially the gorsinol, etc. So certain with using those things only, can we not? Is it not possible to find out the new therapeutics also using the natural product? It's a new molecule itself, for new pharmacological actions.
We can talk about this particular thing.
Hello, sir. So, in one of your slides, I have seen like we can also drive the discovery process by knowing the feature of the active site as well, right?
Right.
So, can we also like target IDPs? Like, as you know, there are a lot of neurodegenerative diseases, which like IDPs are playing a big role in that. And IDPs, for them, the structure is kind of, they have ensemble of structures. So like, is it possible? Like,
I think Chirag has asked about the moonshot problem, and I have keep that part for that portion. Because simply if you think that AlphaFold can predict the structure or deep learning method, but it has not solved the real problem, which is the dynamics, and that's where the IDP question come into picture. I'll talk about during that session.
thank you, sir.
So we need to understand that what we can do and what we cannot do. Yeah. Yeah, I am available, so I can talk to you and others during the coffee break. Thank you.